everyone. I'm Luis Martinez. I'm an associate professor in the Department of Oral Biology at the College of Dentistry at the University of Florida. And the title of my presentation today will be Elucidating the Interactions of Cryptococcus Neoformans and Microglia. So Cryptococcus neoformans is an AIDS-associated pathogen. It has a capsule. It's found in the environment in association with pigeon excreta. It enters into the host by inhalation of the spores of desiccated yeast, and it locates in the lungs. And in the case of immunosuppressions, the fungus have the ability to go into the central nervous system. There, it can cause the hallmark of the disease, which is meningoencephalitis. So Cryptococcus neoformans, there are around 223,000 cases of life-threatening meningoencephalitis, and it can result in close to 200,000 deaths per year due to this disease worldwide. So the fungus has several virulence factors. One is the ability to grow at mammalian temperatures. Also, the ability to produce melanin, which is a pigment that is located in the cell wall of the fungus that protect the fungus against environmental stress, but also have been shown that the fungus can produce this pigment in the central nervous system. Also, however, the most important virulence factor is the fungus polysaccharide capsule. So the capsule is important, it's essential, since a capsular mutants cannot cause disease. The polysaccharide capsule consists of three main components: manoprotein, galactosylomannan of galaxem, and glucuronoxylomannan or GXM, which is the latest is the main component. As you can see here in this scanning electron microscope image, you can see the capsule is very dynamic. As a matter of fact, the fungus can sense the environment and modify the release of the capsule, the production of the capsule during infection. Even it can modify the composition of the polysaccharides or the carbohydrates in that structure. So as I mentioned, it also can be abundantly released in tissues, causing many deleterious effects on the immune system of the host. For example, it can inhibit or prevent leukocyte migration and proliferation. It can affect phagocytosis. It can intervene with antigen presentation. And also have been shown that it enhances HIV replication. As you can see here, you can see that this white material is the polysaccharide capsule of the fungus in a postmortem observation. So that tells you the amount of polysaccharides that are released during infection. So in my lab, we use the intracerebral model of infection. In this case, we use a stereotaxic, which is this instrument, which is connected to a microinjector. And basically, we can infect animals in any region of the brain of our interest. In our case, for this presentation, we selected the striatum, which is a region in the basal ganglia, and it's because this region have been shown to be affected by the fungus, and this region is associated with cognition and motor functions, and both of them are affected during infection. In this case, we infect the animal with 10 to the 4 cryptococcal, and also, it's important to mention that the advantage of this system is that first, as I mentioned, you can specifically infect the animal in specific brain regions. Also, you can control the inoculum. And particularly, you can mimic, depending on the injection, either involvement of the parenchyma or just dissemination throughout the CNS in this type of model. So in this case, we did a first survival. We compare H99, which is a wild-type strain, with a Cas59 strain, which is a capsular mutant. As you can see here, after 10 days, all the animals infected with H99 die. When you check for fungal burden, As you can see here, by day 3 and day 7, you see that the fungal burden in H99 infected animals is higher than in those animals infected with the encapsulated mutant. Also, if you check for the changes in the animal, in the uninfected and the Cas15 uninfected, you can see that the area of the head is very normal. However, in those infected with the wild type strain H99, you can see that there is like a protrusion on the and on top of the head, which is probably because of intracranial pressure in the brain, in the infected brain, but also because of the development of hydrocephalus. If you excise those brains, you can see here again, Cas59 infected, those brains looks very basically healthy, compared to the one on H99 that if you see it very closely, is very like slimy and mushy compared to the other brains. So here, if you do a histological observation or preparation of tissue sections of the brain, in this case is a coronal brain section, you can see that the large cryptococcoma, which is the brain lesion that is formed by this fungus in the brain of these animals. And here you can see all the development of the disease. For example, we can see vacuolated phagocytic cells, or what are known as hueco cells. And they are known like that because hueco means hole in Spanish. And basically, many of these macrophages have vesicles inside of those of the cells. Also, you can see foreign body giant cells, as you can see here. All these manifestations have been previously also described in the human host. Also, if you go into the subarachnoid space, you can see that there are activated macrophages or epithelioid cells. And these cells are usually important in the formation of granuloma, but also, however, they have very minimal phagocytic presence. So if you go in the distal portion distal to the cryptococcoma, you're also going to see ischemic neurons in this the brain tissue. So this tells you the different manifestations that the fungus produce by using this model, but also many of them are also previously described in the human host. Also, as I mentioned previously, the fungus had the ability to release high amounts of polysaccharide. In this case, we have an antibody that is specific to GXM, which is the main polysaccharide component of the polysaccharide capsule. And you can see here in the cortex, you see a lot of, in the brown area is GXM release. Also around the cryptococcoma, you can see a lot of brown regions, which is correspond as you go closer to a lot of GXM release in the area. But also you can, we can stain microglia using a specific marker, which is IVA1. So also, as you can see here, you see a lot of microglia, particularly surrounding the cryptococcoma. Many of these microglia, they look very dystrophic. And basically, we did correlation analysis that shows that the microglia correlate with the intensity or the accumulation of the polysaccharide capsule, in this case, the GXM. So, but what is the role of microglia in controlling Neoformans brain tissue colonization? So microglia is produced in the brain and it stays in the brain. And you can see here in this cartoon, it has a cylindric soma and have many ramifications around the cell. This microglia is important in the homeostasis of neurons. As you can see, it can really release many factors that are important for neurogenesis and also for the maintenance of the synapse. But upon activation, those ramifications retract, as you can see here. And if there is acute or chronic activation there, the microglial cells can express antigen-presenting cell molecules on the surface. And also it can release many cytokines that can be also diminished by the infiltration of peripheral monocytes. On the other side, it can also cause short and moderate activation. And when that happens, it can also release certain factors that could be also deleterious or can cause, can enhance neurodegenerative diseases. For example, Parkinson, Alzheimer's have been associated by overactivation, particularly of microglia. So, but what we know in terms of microglia and Cryptococcus neoformans interaction. From the host side, we know that the capsular material is ingested and localized inside of microglia. As you can see here, these are human microglia with, in red, you see the localization of the GXM. But also in mouse infection, you can see in dark brown, you can see the localization of this polysaccharide. Also, antibodies under opsonizing functions are important for phagocytosis of cryptococci. So, in addition, CD40 and IL-2 enhance the host cell response to the fungus. This is also important because these molecules are usually upregulated by the presence of a very healthy immune system, particularly cell-mediated immunity of the cell-mediated immunity. However, many of these patients with cryptococcosis are immunosuppressed. Also, have been shown that transfer of microglia to control the infections is not effective in protecting the animals. On the pathogen side, there are just a few studies that show the interaction of microglia with the capsule, melanin, and recently we showed the interaction of the capsule with and the importance of phospholipids in the infection, particularly in the brain. This is important because if you go into PubMed and you use the keywords Cryptococcus and microglia, you only find 51 hits for a disease that kills close to 200,000 people a year worldwide, and many people also get infected because of this fungus worldwide. And also it's important to highlight that most of the work done on microglia, most of the bulk of the work was done in the mid-1990s. So we need more people, or more individuals involved in the research of these cells and the interactions with the fungus. So in this case, we use a CX3CR1 mouse, and we can see we infected those mice with Ca59 or H99. Also intracerebrally, as you can see here, there is a heavy recruitment of microglial cells compared to H99. And we were able to quantify these differences by microscopy. Also, you go closer, the cells that are outside of the cryptococcoma, you can see that they're usually branched and ramified. However, as you go closer into this brain lesion, you can see more amoeboid microglia interacting with cryptococcal cells. Also, you can see even rod-shaped type of cells. So recently we showed that microglia can take different shapes during the interaction with cryptococcus. Actually, these are observations that have been seen also in neurodegenerative diseases. However, we don't know specifically what is the role of these different subsets, morphological subsets, in terms of disease, neither in neurodegenerative disease nor in the interaction of cryptococcus with these glial cells. So also in this case, we have different groups, naive, saline injected, H99 and Ca59. As you can see, the animals infected with Ca59, they clear the infection after 3 days. H99, as you can see, there is a formation of the huge cryptococcoma that is characterized by a lot of GXM in the external part of the structure. And when you go inside in blue, you're gonna see a lot of cryptococcal cells, and again in red, you're gonna see the cryptococcal polysaccharide. Also in green, you're gonna see the microglia. So, and this is one advantage of this model, that you can basically trace microglia because basically they're they fluoresce under the microscope. Something that is important here is that during the 3 days after infection, there is a lot of density of microglia near of the cryptococcal that decreases as the infection goes along. As you can see here, after 7 days, most of the cells, they distribute mostly outside of the cryptococcal. If you quantify those cells, you can see here what I just was mentioning, that after 3 days, you have a lot of cryptococcal cells near the cryptococcal, and that reduced after 7 days. If you subtract these brains and you do flow cytometry, also the H99 infected animals show that there is a reduction in the number of microglia compared to the other groups. But also we have the ability to quantify the density and distribution of microglia, as you can see here in the different groups. So one thing that we did is that we determined the density of microglia per micrometer square. As you can see here, the H99 have a higher density of cells after 3 days compared to the other groups. Also, we measure the closeness of neighboring cells. And as you can see here, also in this case, H99 has showed that microglial cells are closer to each other, particularly 3 days after infection compared to the other groups. Also, we determine a region of interest about 12 micrometers and measured how many clusters of microglial cells we can find. And as you can see here, also the H99 infected brains show higher number of clusters compared to the other groups. Then we did the spacing index, which basically is we divide the square of the nearest neighbor index by the microglia density. And again, in this case, we show major differences compared with the naive, but we did not see any difference with the saline injected and the Cap59 cells. So, but also we observe specific changes in the morphology of microglia. And again, here are the different groups. We checked the soma, which is the cell body of microglia, and we did not see major difference, only difference between saline injected Cap59 and the naive. When we check also the arborization area, that is the area where all those branches, of course, and remember when there is interaction with antigen, microglia tend to retract. And we measured that those, the arborization area in H99 infected brain microglia, it was lower than in the other groups. Then we check for the morphologic index, or changes in the morphology, that is dividing the arborization by the soma, and we see that the brains of H99 infected cells have more changes in terms of the morphology compared to the other different groups. But also we use in vitro techniques. In this case, we use one cell line, which is NR9460, which is a microglia cell-like cell, microglia cell. So in this case, we use a transwell in which we place the microglia and we seed the cryptococcal cells at the bottom of the well. The advantage of this system is that they are not in direct contact, but they can transfer chemical signals or molecules through this membrane. And we compare with cryptococcal cells that were just grown by themselves. As you can see here, after those interactions, those cells that were interacting with microglia show that increase in capsule size. When we check for the capsule volume, we see also, again, we quantify that the capsule volume of those cryptococcal cells incubated with microglia have higher, the size is higher. Also, we check for biofilm formation. And in this case, we use the XCT assay. biofilm are a community of microorganisms that interact between each other. And we use the XCT assay. So the XCT is important because basically it measures the metabolic activity of cells within biofilms. So it's a semi-quantitative assay in which the XCT is converted into formals unsolved by the mitochondrial cells of metabolic active cells within the biofilm. And as you can see here, after 24 and 48 hours, we see that those cells that were incubated with microglia show a higher metabolic activity. And we confirm this biofilm information by using viability or colony-forming units assay. And also you can see that there's a correlation with the proliferation and also the metabolic activity in these biofilms. Then we did, we used confocal microscopy. And we compare again H99 cells and H99 incubated with microglia. And as you can see here, the thickness of the biofilm is much higher, higher than when cryptococcal cells are grown by themselves. And we quantify that, and you can see here after 24 and 48 hours, you can see that the thickness of these biofilms are higher than in those cells, fungal cells incubated with the microglia compared to those that were grown alone. And when you take a look closer to those cells, you can see these type of arrangements in under the microscope in India in preparation. You see many cells together after chemical interactions with those microglial cells. So, but also we did, we wanted to test how microglia-like cells respond to direct interaction with H99 and K59. So we wanted to observe what was the advantage of actively producing the capsular polysaccharide. And again, in this case, we did light microscopy, and we measured the distance that these microglia travel after expose them or incubate them with either of the strains. As you can see here, the H99 accumulated lower distances than the untreated and the K59, suggesting that having the polysaccharide capsule protects the fungus from the migration of these microglial cells. But then we check for phagocytosis, and also we see that capsular cells are significantly highly phagocytosed compared to those cells that have the capsule. So basically, these results show that active capsular production by Cryptococcus Neoformans reduces microglia-like cells migration. But also what happened with just GXM, we have the ability, we can extract the polysaccharide in the laboratory. And what we did is that we exposed these microglia-like cells to in presence of GXM or in absence, and we again use microscopy to measure their migration in the field. And as you can see here, also the cells that were exposed to the polysaccharide show lower accumulated distance than those that were untreated. Also, they show lower phagocytosis after incubation for at least 2 hours. And we also observed that the cells that were treated with GXM had diminished capacity of phagocytose multiple cryptococci during those interactions. So this is the model of Neoformans GXM-induced inhibition of microglial migration. So we believe that Cryptococcus get to the brain and begins to release polysaccharide and forms cryptococcomas of biofilm-like lesions. So we're gonna see that outside of those cryptococcomas, many of these cells gonna be ramified, whereas inside of the cryptococcomas, you're gonna see amoeboid or different types of morphologies interacting with the fungus. Also, we, based on the migration assays, we observed that GXM basically inhibit as the infection progresses, inhibit the migration of microglial cells. So in this case of the cryptococcal formation, we have taken some microscopic, this is the idea. So for example, we're gonna have cryptococcal cells surrounded by vast amounts of polysaccharide, which prevent the penetration of microglia and the elimination of the infection. But also, if you see in this case in fluorescent microscope, again, in red, you're gonna see is mostly the cryptococcal cells, and you can see how the microglia are trying to combat the infection during this in the brain. But also, you can see here that you have even closer. You can see that the cells that are inside of the cryptococcoma, most of them are amoeboid or they're activated, as you can see here in the yellow arrowheads. But also outside, you see even when they are very close to the cryptococcoma, you can see that they are these cells are branched and basically non-responding to the infection. So in conclusion, I show that the intracellular infection model and neuroscience techniques can be used to characterize Neoforman colonization of the CNS. We also show that Neoforman GXM is important for fungal brain colonization and mortality. We show that Neoforman infection and GXM release alter microglial responses, distribution, and morphology, and that Neoforman colonization of the central nervous system is mediated by the ability of the fungus to form cryptococcomas and failure of the microglia to control the infection. And why this work is important? So it's significant because understanding the cerebral cryptococcosis is critical to prevent and develop novel therapies for the treatment of the disease. With this, I would like to thank all the people in my lab, particularly most of the work was presented in this presentation was done by Vanessa Enriquez that just recently left the lab to accept a position at Emory Medical School. Most of the migration assays were developed by Melissa Munson. She's a very talented graduate student in the lab. The incorporation of the intracerebral model was done by Mohammed Ahmed. He's currently a faculty at the University of Mansoura in Egypt. And also Claudia Charles Niño and Marta Reguera Gomez both were very important in terms of supporting many of these experiments. Also, I would like to thank my collaborators, Dr. Karina Albina here in the Department of Neuroscience at the University of Florida. She was very instrumental for us to develop the intracerebral model and all the setup. And a longtime collaborator at Hofstra University, Michael Dors, as well as Ada Lee at the New York Institute of Technology, College of Medicine, who help us in the initial studies with bioinformation. In addition, I would like to thank the funding by the National Institute of Allergy and Infectious Diseases, and also Vanessa, particularly Vanessa Enriquez, was supported by a training grant for the NIDCR here at in the Department of Biology here in the College of Dentistry at the University of Florida. And with that, I thank you for your attention, and I will be in the lookout for your questions. Thank you very much, and have a wonderful day.